Semester I Human Anatomy and Physiology I Handwriting Notes Unit -1 Chapter 2 on Chapter 2nd Cellular Level of Organization Syllabus 1 Structure and Functions of Cell Transport across cell membrane, cell division, cell functions. 2 General Principles of Cell Communication Intracellular signaling pathway activation by extracellular signal molecule Forms of intracellular signaling A. Contact dependent B. Paracrine C. Synaptic D. Endocrine. This is the cellular level of body, which contains cells, tissue, organs, organ system, body. Molecular level? Cell. It is a structural, basic, and functional unit of our body, all living organism. A human body consists of about 10 nanoplans or 100 trillion cells, with a size and mass of about 10 micrometers and 1 nanogram, respectively. Cells are of two types. Prokaryotic cells. These are those cells which are not fully developed. They have less developed nucleus and some organelles, e.g. bacteria, etc. Eukaryotic cells. These are those cells which are fully developed. They have well-developed nucleus and other organelles present in cell, e.g. plants, animals, fungi, etc. Cell. A cell is the basic living structural and functional unit of body enclosed within a membrane. Fear diagram just may labeled, hey? Cytosol, endoplasmic reticulum, ER, nucleolus, Golgi apparatus, lysosome, centrosomes, plasma membrane, cytoplasm, ribosome, nucleus, peroxisome, mitochondria. There are about 200 different types of cells in our body. Functions of cell, movement of substance across the cell membrane, cell growth and metabolism by cell division, protein synthesis by transcription and translation. Structure of the cell, it is divided into 3 major parts. I plasma membrane, I cytoplasm, and I nucleus. Plasma membrane, also known as cell membrane, it is the outermost membrane of cell which separates out the internal environment of cell to the external environment. It is selectively permeable, which allowing substances to pass through it. Natural structure. The fluid mosaic model of plasma membrane was given by S.J. Singer and G.L. Nicholson. It is made up with bilayer of phospholipids. Chemical composition. Phospholipid, phosphate plus lipids, phosphate hydrophilic, lipid, hydrophobic. Proteins. The lipid bilayer is embedded with proteins of various shape and size, e.g. integral, transmembrane, and peripheral proteins. Carbohydrates. These molecules are attached with proteins, glycoprotein and lipids, glycolipid. Functions. It provides protection from external environment. It gives shape to the cell. It regulates the flow of material into and out of the cell. It also plays a key role in communication with other cells or external environment. Cytoplasm. It is the total area between the plasma membrane to the nucleus. It further divided into 2 parts, 1 organelles, 2 cytosol. 1 cytosol. It is the fluid present inside the cells. It is transparent, viscous, which contain about 75 to 90% water, protein, lipids, and carbohydrate. 2 organelles. These are the organelles present in cytoplasm and perform specific function for cell. Endoplasmic reticulum, ER, Golgi apparatus, body, complex. Ribosomes, mitochondria, lysosomes, centrosomes, peroxisome. Endoplasmic reticulum, ER. It is a complex channel system enclosed within the membrane in the form of 3-dimensional network that consists of vesicles, flattened sacs, and branched tubules. Nietzsche diagram jessa nucleus, cisternae, smooth, rough, ribosomes. It is of 2 types. I, rough ER, those which have ribosomes attached, helped in protein and membrane synthesis. I, smooth ER, ribosomes are not attached. Functions. Act as a structural framework of cytoplasm, exchange materials, SER, help in synthesis of phospholipids, cholesterol, and triglycerides. Ribosomes. Ribosomes, these are made up with 2 subunits, i.e. smaller subunits, 40s, and the larger subunit, 60s. 2 types, one is attached with ER and one is present in free form. These are tiny spheres that contain ribosomal RNA and several ribosomal proteins, also known as factory of proteins, because protein synthesis takes place in it. Diagram, 40s plus 60s equals 80s. Amino acids, mRNA, protein. Mitochondria, it is made up with 2 membranes, i.e. inner and outer. The main function is to generate energy, also performs cellular signaling and regulates cellular proliferation. Spread, it is known as the powerhouse of cell because it generates ATP. Diagram, inner membrane, outer membrane, intermembrane space, cristae, FOF1 particles, mitochondrial matrix, DNA, ribosomes. Golgi apparatus body. It consists of 4 to 6 flattened sacs called as cistern placed upon each other, like a pile of plates with expanded bulges at their ends. Upper Vale diagram में, cis region, medial region, trans region, cisternae, lysosomes, vesicles. Functions. They stores proteins, modify them, and also move to the plasma membrane through secretory granules when required, also helping in excreting excess amount of water. Lysosomes. They are secretory formed from the Golgi complex. They worked as the digestive system of cell, which contained digestive and hydrolytic enzymes and hydrolyze large molecules such as RNA and DNA, proteins, and lipids. Vary in size, ranging from 0.1 1.2 micrometer. They also work as defending against microorganism. Centrosomes. It consists of a pair of centrioles and play an important role during cell division. Centrosome. Diagram. Centriolus. Peroxisome. It contains many oxidases enzymes that can oxidize various organic substances such as fatty acid, amino acids, uric acid, etc. It contains the enzyme catalase and protect the cells against toxic effects of substances. Diagram. Lipid bilayer, crystalline core, enzymes, transport protein. Nucleus. The nucleus is usually a spherical and oval-shaped structure, which is largest in the cell. It consists of double membrane, which separate nucleus from the cytoplasm. It contains a spherical structure called nucleolus. It is also known as the control center of the cell. Diagram. Nucleolus, chromatin, nuclear pore, nuclear envelope, nucleoplasm. It contains the genetic material including aggregation of protein DNA and RNA. It directly involves in the reproduction and transfer all genetic information from parent cell to daughter cells. As written, transport across membranes. Plasma membrane of cells are selectively permeable, which allow only some substance to cross it. Diagram. Not allowed. Plasma membrane. Third image. Membrane transport. As written. Membrane is a biological membrane, which is made up with phospholipids bilayer and allows substance to move through it. It is known as membrane transport. Substances are transported across the cell membrane through I. Passive transport, passive diffusion, facilitated diffusion. I. Active transport, primary transport, secondary transport. EI. Endocytosis. Concentration. As it is written. Concentration gradient. When any drug substance move from high concentration to lower concentration. Yada se kam against concentration gradient, kam se yada, chik ke niche. High amount, low amount across const gradient. I. Passive transport. In this, substance transport across the constant gradient, i.e. high to low. Passive diffusion. It is the transport of substance across the constant gradient, i.e. from the region of higher cons to lower consine without use of energy. E.g., diffusion of lipid-soluble molecules like O2, CO2. CO2 across the cell membrane, diagram Milikoa text. Extracellular fluids membrane. Intracellular fluids for weak soluble drugs pore, any channel. Filtration, AQ diffusion, high to low through AQ protein. Facilitated diffusion. In this, substance transport across the constant gradient, but with the help of any carrier bodies. That's why it is also called carrier-mediated transport. It is suitable for poorly diffusible substance carrier used, such as SLC, solute carrier transporter, e.g., entry of glucose into RBCs. Intestinal absorption of B1 and B2. Diagram text of carrier bodies. Drugs carrier bodies, e.g., SLC. Osmosis. It is the movement of solvent water particle from high to solvent low consane through semi-permeable membrane, from low solute cons to high solute consane. Active transport. In this, substance transport opposite against the constant gradient with the help of energy. In this, ATP is used. ATP, hydrolyzed, ADP. It is of two types, primary active transport, secondary active transport. Primary active transport. In this, ATP is used as an energy, e.g. absorption of glucose, e.g. NPK ATPase pump. 2. Secondary active transport. In this, electrochemical gradient are used instead of energy. Symport cotransport, movement of both substance in same direction, e.g. NMI glucose symporter. Antiport countertransport, movement of molecules in opposite direction. Endocytosis. In this transport, drugs of very large size molecules get transported via engulfment by cell membrane, e.g. cellular uptake of macromolecular like fat, starch, oil-soluble vitamins like A, D, E, K, and drug like insulin. Now page 5, diagram headings exactly as written. Outside macromolecule process of engulfing, inside cell membrane, macro drug. Phagocytosis, cell eating, adsorptive uptake of solid particulates. Pinocytosis, cell drinking, uptake of fluid solute. Cell division. It is a process by which a parent cell divides into two daughter cells. The genetic content, DNA, of newly formed cell is exactly same to the parent cell. Page 3. Types of cell division. Cell division is of two types. I, mitosis, responsible for growth of cells. No. Of chromosome same equational division occurs in somatic cells. I, meiosis, responsible for formation of gametes cell occurs in reproductive cells, and no of chromosome decreased, reductional division. Page 4. Cell growth sketch increases the no of cells, i.e. amount of cells, parent cells shown splitting into two identical daughter cells. Page 5. Cell cycle definition. Cell cycle, the sequence of events by which a cell duplicates its genome, synthesis the other constituents of the cell, and eventually divides into two daughter cells. It has two phases. I, interphase, preparation, M phase, cell division. Of text in diagram. GZO phase, first growth phase, cell growth, ATP, nucleotide RNA and protein synthesis, preparation for DNA replication. S phase, synthesis phase, DNA replication, DNA duplication, replication of chromosomes, centriole duplication. G2 phase, second growth phase, RNA and protein synthesis, preparation for mitosis. M phase, cell division, prophase, metaphase, anaphase, telophase, cytophase, or resting. 1. Mitosis, also known as M phase or mitotic phase. Equational division. It occurs in somatic cell in which already existing parent cell divides into two identical daughter cells. In this, the No of chromosome in daughter cell is same as the No of chromosome in parent cell. Page 2. It occurs into two parts. I, karyokinesis, prophase, metaphase, anaphase, telophase. T, cytokinesis, diagram showing 2N, 2N cells. At page 3, I, karyokinesis. It is a process by which the cell nucleus divides into two daughter nuclei. Diagram of nucleus dividing. Now page 4, prophase. This phase begins with initiation of condensation of the chromosomal material, i.e. chromatin. Centrioles, centrosome, move towards the opposite poles of the cell. Organelles like Golgi complex, ER, nucleolus, and nuclear envelope disappear at the end of prophase. Diagram with nucleus, centrosome, mitotic spindle fibers. Haber page 5. Metaphase. This phase begins with complete disintegration of nuclear envelope. Chromosomal condensation completed and two sister chromatids are held together by centromere. Kinetochores, small disc shaped at the surface of centromere. Spindle fiber attached to kinetochores and chromosome are moved to spindle equator and get aligned. Diagram of chromosome alignment. Page 6. Anaphase begins with splitting of each chromosome. Separation of chromatids and move towards opposite poles. Diagram of separated chromatids.
वीडियो अच्छी लगी हो तो प्लीज़ लाइक कमेंट एंड शेयर एंड प्लीज़ सब्सक्राइब।